Good morning, afternoon, or evening, depending upon where you're joining from. My name is Heather Stark. I'm the Global Segment Market Manager covering biopharma for Oxford Nanopore. Today, I'm joined by Dr. Andrea O'Hara from Genewiz to talk about enhancing cell and gene therapy success with multi-omic sequencing, the Oxford Nanopore Advantage. So I'd like to provide a quick overview of nanopore sequencing. There are 4 key components that enable this technology. A motor enzyme is attached to DNA or RNA molecule via an adapter, which binds by either ligation, tagmentation, or PCR if needed. In the presence of an ionic current, the motor enzyme plays a crucial role by controlling the movement of the strand through the nanopore, unwinding and feeding it through the pore, which is embedded in a non-ionic supporting membrane. And this happens at about 400 bases per second. As the nucleotide passes through the nanopore, it causes a distinct disruption in the ionic current. These disruptions are detected and recorded in real time, allowing the specific sequence of nucleotides to be determined based upon pattern recognition, a pattern recognition algorithm that's running on a recurrent neural network. So these pores are arranged in arrays, which enable massively parallel sequencing, which is not dependent upon fluorescent nucleotide incorporation. So no dyes, lasers, or pictures are needed after each incorporation. The more pores on the flow cell, the faster it sequences and the more data it generates. You can see that there are about 4 to 5 times the number of pores on each flow cell. And due to this novel nature, there are several inherent benefits. So we can directly sequence native DNA or RNA. This eliminates the need for amplification or reverse transcription, although it's still an option. This eliminates PCR bias, and it generates rich data sets that encompass epigenetic information as well as base qualification. There's minimal sample preparation. Our fast and easy library preps reduce hands-on time, but they also make Nanopore read length agnostic. Imagine the ability to sequence everything from tRNAs to entire genes to large genomic regions like centromeres in a single contiguous read on a single platform. This improves assembly and structural variant detection, and it's a more accurate representation of the libraries. Nanopore sequencing is also in real time, which allows for immediate access to results, facilitating quick decision-making and novel applications like adaptive sampling, which is a library prep-free enrichment using bioinformatics during the sequencing run. And lastly, we offer cost-effective and accessible chemistry that scales across a range of devices, which Genewiz has. This enables easier feasibility in large-scale screening studies. So overall, nanopore sequencing offers a flexible, cost-effective, and rapid approach to sequencing that's revolutionizing genomics research. Now that you've seen how nanopore sequencing works, I'd like to discuss a few of our end-to-end workflows that could be utilized. Most gene therapy starts with plasmid construction. We recommend nanopore whole plasmid sequencing to verify not only is your insert free of mutations and in the right orientation, but also that your vector backbone is free from deletions, dimers, or other mutations that can affect delivery. Moving over to viral vectors, Nanopore's developed a method for sequencing AAV. These single-stranded, positively and negatively charged genomes self-complement in nature. So this provides enough double-stranded structure through their ITRs that we can easily add adapters without much upfront library prep. There's also no heat, denaturing, or annealing required, as we found this leads to a prevalence of smaller fragments. We also don't recommend a size selection during bead cleanup to get a true representation of the library. So you can pair this with our simple epitome AAV QC library analysis, and to not only detect the transgene, but to distinguish between full length and partials, and to capture the full construct ITR to ITR. So you can see those complex events, identify hotspots in the ITR, as well as detect contaminants. So moving over to RNA vaccine development, after you verify the identity integrity of your plasmid constructs, we recommend using Nanopore's direct RNA kit for the in vitro transcribed RNA intermediates. You can capture the mRNA in its native form from 5' to 3' UTR, as well as the transgene and the complete poly A tail with modifications, allowing you to distinguish between full-length poly A and truncated species. This takes about 4 hours of sample, and you can use our workflow mRNA QC for this. For discovery work, identifying novel antibodies or profiling your CHO cell lines are animal models to evaluate where your target genes of interest may have inserted in the genome, you could consider nanopore whole genome sequencing with methylation to ensure that there are no hyper or hypomethylation, for example, in promoter regions that could affect therapeutic protein expression, as was done by Dr. Winston Timp at JHU. You might consider complementing this with single-cell sequencing from Nanopore, which is compatible with 10x 3' and 5' NextGem libraries to generate full-length isoforms, gene fusions, and SNPs to further verify allelic expression for complete cell characterization. This prep generates around 80 million cell-assigned barcoded reads, and our analysis provides gene annotation and expression count makes, as well as other quality metrics. Thank you for your time and attention today. I'd now like to hand it over to Dr. Andrea O'Hara, who's a multi-omics expert from Genewiz, who will discuss how to use nanopore sequencing technology to enhance the precision and efficacy of cell and gene therapy programs.
Thanks so much for that introduction. Before we begin, I'd just like to introduce myself a little bit more. So I am coming at you from Genewiz from Azenta Life Sciences. Azenta Life Sciences is actually a union of a bunch of different companies all together. This includes the Genewiz synthetics and multi-omic solutions, as well as a number of other different commercial entities. These all focus primarily on sample management and storage solutions. This includes things like on and off-site storage, cold chain solutions, as well as consumables and instruments. If you have any questions about any of these other product portfolios or about the Genewiz solutions, my contact information will be at the end of the talk. But for today, I'm really focusing specifically on the Genewiz multi-omics and synthetic solutions. So if we think back to the previous presentation that we just saw, and that cell and gene therapy development pipeline, there's a number of different ways that we can utilize Oxford Nanopore sequencing throughout the workflow. This is a kind of a high-level view of the workflow, and I want to just take some time to go through it, and then we're going to dive in a little deeper. So the upstream part, we're going to focus on discovery and identification. This is really the basic research part where we're going to be looking at identifying that sort of area of interest that we want to study. So we're going to start out by taking our disease samples, whatever the disease may be, identify mutations, validate targets, that target discovery phase. Once we've identified the particular targets, then we're going to start to create libraries of constructs and identify leads for the therapy. So this is therapeutic lead identification. This screening process will take us into our actual candidate leads. This can be one, it can be many. Depends on what kind of tool we're using, portfolio, what we're looking at. But at that point, once we have a smaller number of candidates, that's where we're going to start to optimize this. We're going to, if we're doing viral vectors, we're going to put them into the vectors, optimize our vectors, and really hone in on that processing. And once we have our best version or versions, we're going to then produce the product. At that stage, we're also going to do our preclinical testing. That's going to be things like cell line testing, animal testing, and maybe even a small number of patients. From there, we're going to go into the downstream clinical trials. That's where we're going to look at our larger cohorts. We're going to stratify our patients. We're going to do larger clinical trials. We're going to manufacture and scale up and bring this, to the big picture. So for today, we're going to really focus primarily on this preclinical developments and also a little bit on the end of that lead identification part, but thinking about how can we streamline this using Oxford Nanopore as a means to optimize our accuracy, efficacy, and safety. Safety is always number one, but in order for us to have that safe product, we have to make sure that it is accurate, that it is correct, and that's where that sequencing is going to come into play. So for our first example, we're going to use AAV. I'm going to take you through two different examples where we can use this sort of system. So if we're thinking about AAV, we want to think of the general viral packaging workflow. This is a high-level view of that packaging workflow. We're going to start out by designing that construct, sequencing it, and synthesizing it and sequencing it so that we end up with our plasmid. Once we have a plasmid, we're going to put it into our viral vector. Again, here we're thinking AAV. then we're going to actually determine the titer and make sure that our product is correct. And we're going to zoom in on these parts in a little more detail. So if we start out with that upstream part, how do we create that first plasmid that's going to take us to our final product? For AAV, we have two different methods of gene synthesis that we can use to actually create our plasmid. The first is linear DNA synthesis, and this is where we're just going to synthesize the fragment that's going to fit into an already existing plasmid backbone. The other option is to actually synthesize the entire plasmid end to end. Either of these options are fine. Which one you choose is really up to you and your preference. But the thing we want to keep in mind here is that we have this really fantastic, perfect AAV transfer plasmid, and it's going to have some key factors. So first off, we're going to have the backbone. That's that bottom part shown in gray. And then when we are actually making the plasmid, we're going to have our ITR regions, which are shown in purple on the left and right, and our insert. The final AAV product is this very unique sort of barbell structure I'm showing on the bottom right. And you can see we've got the ITRs on the ends and the transgene. It's really critical that all of these parts are perfect, that the ITRs, both left and right ITR, and the transgene are perfect. The transgene is what we think of as being what's being delivered. It's obviously, this is the part that we're focusing on. We've done all of the research to generate it. So we know we want that to be correct. But the ITRs are equally as critical. We need to make sure that they have a perfect sequence, that there are not any insertions, deletions, or mutations. ITRs are really, really, really tricky. They are prone to mutation and they're difficult to sequence, which means that we really need to have a great solution to test it, to make sure that we've maintained that fidelity, and something that's going to be able to read through that secondary structure. So when we're looking at that transfer plasmid, we want to make sure that those parts are all correct. We also do want to make sure the backbone is correct. Even though it's the backbone and we think of it as being less important, it's still going to have some really critical functions. And if there are errors in the backbone, it can impact that downstream product. So we want to look at this holistically. So to do that, we can use Oxford Nanopore sequencing. At Genewiz, this product is called Plasmid Easy, but you can think of Oxford Nanopore sequencing in general. Oxford Nanopore is really ideal for complete plasmid sequencing because that long read technology allows us to sequence this plasmid in its entirety. The first step we're going to do is we're going to linearize the plasmid randomly. This is important because we're not going to get any bias. So we're going to linearize the plasmid and we're going to have a pool of plasmids. So we will have, hundreds, thousands, millions, however many we want. And then we're going to sequence each of those plasmid pieces all the way end to end. What this means is that we're going to be able to determine that our backbone is fully intact, looks correct. We're going to make sure that we have both ITRs and that we have our insert. And we'll be able to make sure that this is a totally clonal population and that our ITRs are perfect and our transgene is perfect. We're also going to sequence this at a depth to make sure that it's clonal and we don't have any variables. Oxford Nanopore in particular is really ideal because this is a very simple, straightforward workflow, and we can get these results overnight. So you can submit a plasmid and get the sequencing results the very next day. So this is perfect for your upstream generation, both for screening as well as when you have a more mature product, but a perfect test for us to test that entire plasmid. Now, once we've confirmed that our plasmid is perfect, we need to package this into the viral vector. And again, here we're thinking that AAV vector. So what does this look like? We're going to start out with that transfer plasmid, and then we're going to incorporate, two additional plasmids, the rep cap and helper plasmid. All three of these are going to be transfected into that host packaging cell line. Typically, this is a human cell line. It doesn't need to be, but it usually is. And then from there, we're going to harvest the AAV particles. These are going to be the packaged recombinant AAV. And again, within that is going to be that recombinant AAV DNA, which is that barbell with the transgene. Now, when we're thinking about the packaging, if everything goes according to plan, all we'll have inside of those AAV particles is that recombinant AAV DNA, but there is the potential for us to have other contaminating DNA. This can be things like the backbone of the transfer plasmid, the rep cap or helper plasmids, or any DNA from the host packaging cell line. So it's really imperative that we test our product, that packaged product, those AAV viral particles, to make sure that we have exactly what we expect, no more, no less. So once we've completed the packaging, we're going to determine the titer, and then we're going to confirm our viral products. And to do this, we're again going to think of our next generation sequencing, and in specific, that Oxford Nanopore technology. Now, with this downstream sequencing, we're going to be sequencing that recombinant AAV product, and we're going to be thinking about three main factors here: the purity, the clonality, and the fidelity. With the purity, we want to make sure that we're able to see our recombinant AAV DNA and no other products. That's going to be, that backbone, two other plasmids, or any of that packaging cell DNA. Clonality is we want to make sure that our ITRs and transgenes are totally correct. We don't have any sequence variants. There was no mutations that cropped up during this whole development process. And then fidelity is making sure that it's intact in terms of its length, so that I've got the full left ITR, transgene, and right ITR. And we don't have any potential truncation events, loss of an ITR, shorter ITRs, or if the transgene went in and didn't go fully in. Again, we should have already been checking our plasmids upstream, so there shouldn't be as many surprises here, but we just want to make sure that that packaged product is correct. So to do this, we're going to take our AAV particles, we're going to extract that DNA, and then we're going to sequence it. Now with this one, unlike with the plasmid sequencing, we're not going to do any fragmentation because I want to keep that DNA intact. Ideally, it is just that recombinant AAV DNA. And we're going to be able to load this onto the Nanopore sequencer and get our direct results. Now, when we do that direct DNA sequencing, we'll be able to determine if we have a full-length genome. We're going to be able to see, are there any variants, whether they're in the ITR regions or in the inserts. We can also identify if we have any truncation events or if it's fully intact. Depending upon how deep we sequence, we can also determine if there are any contaminating products. And again, this can be backbone, other plasmids, or that host cell DNA. We can also determine if we've had anything sort of really wild and wacky happening, chimeric events, things like that. Because we're sequencing the whole molecule, whatever that molecule may be, we'll be able to see exactly what we got. Now, because Nanopore sequencing is a very straightforward, simplistic workflow in terms of getting that prep done, and it's a fast sequencer, it's going to be really ideal for screening both early and late stage products. In the early products, when we're just making those first versions in that sort of earlier R&D phase, we might sequence less deeply and really just focus on fidelity of product, making sure that I'm getting that construct in there and that it looks good. Once I have a more mature product, I'm typically going to want to sequence it deeper so that I can confirm that I already know I have this good product, but that I don't have any other contaminating effects. But the Nanopore tech is really ideal for both stages. Now I want to switch gears a little bit here and think about other products that we can use. So in terms of gene therapies, lentivirus was sort of our tried and true. AAV is the up and comer, but it's very, very prominent. Our new kid on the block here is mRNA therapeutics. And similar to AAV, we can use mRNA therapeutics. We can use the ONT workflow for mRNA therapeutic development. So I want to take a deeper dive as to what this would look like for an mRNA therapy. Now, with the mRNA therapies, just like with AAV and any other therapy, we want to make sure that we have our perfectly pristine synthesis upstream, and that we're developing a perfectly pure product downstream. So for both of these, any way we slice and dice this, we want to make sure that we are sequencing frequently and that we're getting that correct result. So to take a look at this workflow, it's going to look really similar to the AAV workflow we saw before. We're going to start by identifying that mechanism of disease, and then we're going to identify our treatment target or biomarkers that we want to hit and create the therapeutic. Once we've created a single therapy, more likely many therapies, we're going to screen them to identify which one looks the best. Then we're going to adjust the dosages, identify our delivery method, and then we'll start our preclinical studies against cell lines, animal models, small cohort of individuals. And then finally, we'll move into our larger clinical testing. Now I want to zoom in again on that creation of the therapeutic, as well as that screening. So if you look here at the bottom of your screen here, you're going to see at the very bottom, we start out actually with our DNA sequence, and we're going to do gene synthesis at the DNA level to start. I know we're creating an mRNA therapeutic, but we're starting with DNA. And when we make that first gene synthesis, we're going to be thinking about our upstream 5' and 3' UTRs, as well as the poly A tail length. And we're going to want to transcribe that we're going to synthesize that entire product, and then clone that into our plasmid. Again, coming back to the plasmid. The plasmid will have our backbone as well as our T7 promoter region, and we're going to put that all together, and then we need to sequence that plasmid to make sure it's intact. Again, we're going to be using our Oxford Nanopore plasmid easy sequencing, where we just submit the plasmid and we get those results back overnight. Here, again, random fragmentation, single fragments, so that we still get a long piece. But we're going to be looking for slightly different structural components. So with this version, since we're looking at the mRNA therapeutic, we're going to focus in on making sure that we have that nice clean backbone, that we have our intact promoter region, and that we have the 2 UTRs, our region of interest, and then that poly A tail. We really want to make sure that our poly A tail length in particular is very good. It can be too short or too long, and both of these things, too short or too long, can really impact how long the molecule stays around in the cell. Is it effective? How well is it translated? So many factors here. So poly A tail length is really critical. Again, we want to make sure, though, that our region of interest is also fully intact. Now, once we've confirmed that we have this perfect plasmid sequence, we're going to scale up. We're going to linearize it, do our in vitro transcription, and we're going to actually generate now that pool of either linear or circular mRNA. It depends on what your flavor choice is here. And then we need to QC it again to make sure that that product is correct. Now here, at this stage, we have an mRNA. Now what's super amazing about the Nanopore technology is that we can do direct sequencing of the mRNA. So we can take this mRNA and we can determine the same types of things we were looking at before with the AAV. So that clonality, fidelity, and making sure that we have a full-length product. So we're going to do a full-length mRNA sequencing. Again, we're not going to do any fragmentation. We're taking that full product and looking at it end to end. We'll make sure that we've got everything at the very front, that we didn't get any premature cropping upstream, that we have the whole molecule, and that we have the full poly A tail. We're also going to be able to calculate the abundance of variants. There's always the potential that some sort of point mutation could have cropped up, so we want to make sure that that's not present. But we can also determine if we have a polyvalence combination. Some mRNA therapeutics are going to be monovalent, meaning there's just one version present. Some might have multiple variants. And so we can actually use this abundance approach to make sure that they're represented in the correct proportion. So do I have 30, 30, 40 or 33, 33, 33? And again, what version you want is going to be up to you, but you'll be able to determine that with your next generation sequencing. And then lastly, we're going to still make sure that it's all the way intact. So we want to make sure there was no truncation events. Is my poly A tail length correct? Does it have a tail? Did I get products without a tail? Again, just like earlier, we can use ONT for both early and late stage screening. And how much we sequence this here will kind of depend what we're looking for. So for those earlier upstream products, we might be doing a little bit less sequencing and focusing on a larger cohort of products to sort of narrow down. And then once we're in the later stages of development, we're going to plan on sequencing deeper just to make sure that we have everything very robust. But again, thinking about sequencing all the way across stage after stage. Now, once we've created our product, whatever it might be, we're going to want to take this into our clinical trials and use them within the larger clinical trial. Now, in the clinical trial, that's going to exist for a very long time when we go to market. And I want us to now switch back and think a little bit about AAV and where we can use Oxford Nanopore in particular for some of the downstream clinical trial testing. So integration site analysis, or ISA, is where we are going to check host genome for potential integration. ISA testing is required by the FDA for all viral vector treatments, both AAV and lentivirus. Lentivirus is meant to be integrating AAV, typically as a non-integrating option. But for either approach, we do need to still test to see if the viral vector has integrated, do we have that integration, and where the integration is. When you test this will depend on which method you have here, AAV versus lenti, but both have FDA requirements. Now, for ISA testing, there are a whole bunch, huge number of different methods that you can use, both targeted and non-targeted approaches. There are pros and cons to either one. Some of it will depend on which viral vector you're using. Do you have expected integration sites? And what is your goal? What are we looking for? So which targeted or non-targeted approach you use is something that you should definitely have a consultation for to learn the details. But regardless of which one you pick, targeted or non-targeted, we're still going to be looking for some of the same approaches here, which is to identify, is there an integration? Where is it integrated? And how many times is it integrated? Long-read sequencing is fabulous for this because it will let us know if I have multiple integrations in a row, because I can sequence that whole long molecule. So I'm going to get a larger part of the DNA hits. It will be able to allow us to test dark regions that might be really difficult to test with short-read technologies. And depending upon method, we can do non-targeted approaches that are going to be very unbiased. So we'll get a very holistic view of what's going on. But in general, long-read sequencing with Oxford Nanopore is really ideal for ISA testing. Now, we know that you have a lot of different partners to choose from for both your Oxford Nanopore and NGS sequencing as well as synthesis, but we hope that you choose Azenta. We are the trusted partner for over 4,000 institutions, including all top 30 pharma companies, and we have 30 locations globally around the world. So we're really here to support you with whatever you might need. And if you do have any questions or want to reach out to get some more information, you can either email directly at ngs@azenta.com, or you can scan this QR code, and that will take you to our Contact Us page.